What is the fungicide? So I mean, say any molecule, either it is plant origin, either it is a chemical origin or synthetic origin. If they are able to kill the fungus, they are named as fungicide. So I think most of you are clear that fungicide are the molecule those who used to kill the fungus. Why the fungicide it is required in the agriculture system, in the public health system, in the veterinary system? Because a large number of the diseases they have been caused by different kind of fungus in the human being, in the plant, as well as in the animal and other amphibians also. Recently, I think you might have uh, come across a news that a lot of frog and many amphibians they have been died in their natural ecosystem because of the a new fungus that has invaded to that all those amphibians, and it is a great catastrophe for the wildlife. As far as agriculture is concerned, we are the biggest consumer of the fungicide, and fungicide it has been used and it has been introduced in the agri ecosystem in around 1845 by Professor Millardet. It was an accidental phenomena when he was walking from the Bordeaux University. He was walking through the grapevines and he has observed that the grapevine, those have been sprayed the copper oxychloride, and that wine, it has free from the infection of the downy mildew. But farmers, they have used the copper fungicide or the copper formulation not to control the fungus. They just to control the animal and other insect, those who are coming and eating the leaf. So this was the accidental discovery, and a fungicide, chemical fungicide, came into existence. If you look about the Indian system, so mostly we used to trace the use of these chemicals, and it has been documented in the Briksha Ayurveda. In the Briksha Ayurveda, for the promotion of the crop health and good production, farmers they tried many things, like they used to use the jaggery, they have used the ghee and then fermented milk, cow dung, many preparation, and they use some to up to some extent they used to control the fungus and diseases. But the real revolution in the formulation of the fungicide, it came after the discovery of the Millardet, and since then we have realized the power of the fungicide for the control of the many plant diseases. So I am going to my first slide. So you will see this is the infection of the but the pigeon pea as well as the wheat crop by the fungus, and it is going to incur a loss of around 70 to 80%, because most of the plant they are going to be die. If some plant they are going to be survive, the their ear, their fruits, it will be very small, and in some cases plant will be there, but fruit it will not come. So the I mean, so that around 25% crop produce it is going to be lost because of the various fungal diseases. So it is a great loss. If you will calculate the globally, it is going to, I mean, say, create the loss of the several $100 billion. And many times this loss, it is going to create the kind of famine. I'm just coming. Just this slide I'm just producing before you, that everyone is remembering that the Great Bengal famine that has occurred in India in 1943. This Great Bengal famine, it occurred only because of the outbreak of Helminthosporium in the rice. Rice is the most stable food in the United Bengal province, and there this crop it has been failed, and there was a shortage of the food material. Fortunately, that in India, Punjab and Haryana, they had enough food by the time it was United Province, Punjab, Haryana, and both they were all together. But because of the Britisher intervention, the food supply could not came from Punjab. Of course, rail and everything it was there, but the food could not be supplied to the Bengal, and millions people, 2.1 to 3 million people, they have died because of the starvation or starvation related diseases. So it was a great. Then government of India and appointed a royal commission for the research in the agriculture. I would say that this thing, it has uh, started, and government has given more emphasis about a new kind of agriculture, and so that the food security could be improved and the losses caused by the diseases and pests, it could be minimized, because Bengal famine, it was resulted only because of the uh, outbreak of a fungal disease that is called Helminthosporium. So the same thing, fungicide play the crucial role in protecting the crop from the fungal diseases, and changes in the farming technique and technology advancement have encouraged the farmer to effective the pest management. See why the farmer it is coming for the pest management. The pest management is a very, I would say, a kind of managerial skill. It is very simple. If I am investing 1 rupees, I will continue the business if I will get at least more than 1 rupees. So every investment, how much earning we are getting. So all the technique and technology, those who are promoting our benefit ratio, we are adopting in the agriculture or any kind of business. So fungicide that we are using here in the agriculture ecosystem, they are ensuring our crop, our better produce, and our increased income, our increased profit. That's why farmer they are purchasing the fungicide from the companies and using in their field, in their farm, just to get the good quality of produce, just to get the higher yield, just to get their better income. So, so now if you see about the global fungicide market, it is around 13.4 billion. It is a huge and expected to reach around 19.5 billion by 2027. So this is the, I would say, you can imagine this business. And what I am talking about this business is mostly dominated by the synthetic fungicide, mostly the chemical fungicide. Now this, now I'm coming to my topic. What is the problem? Why we are thinking about the biofungicide? Of course, we have started from the biofungicide. If you look the Vriksha Ayurveda, if you look our ancient and I mean say medieval, if you look about the history of the agriculture in the Middle Ages and in the beginning of the agriculture, we mostly used several bio-based product, but it was a crude product and it was not given any scientific explanation. But still, it is needed. So the biofungicide, they have their some specific and unique feature, and they can give the better advantage. The reason was very simple. Number one, it is a long-term goal for the productivity could be achieved. See, the fungicide which we are using, we are having several problems with those fungicide, and no or minimum residue. Residue is the most, hello, residue is the most important problem. If you are using any chemical in agro ecosystem, it is going to persist for the longer period, and it is going to damage the many microflora, fauna, and the residue it is coming in our produce, and then it is going to be, I would say, human system, animal system, and water system, and ultimately it is creating problem of the health, problem of the environment. I mean, there's so many environment and health related issue it is creating. But when we are going to use the biofungicide, definitely I'm not giving guarantee, I'm not sure that it is going to be give a clean environment, and it is going to support our health, but definitely it will be better. What it will be 1,000 times better than the synthetic or the chemical based fungicide. No or minimum damage to the environment, that already I have discussed. And benefits should be extended to both producer and consumer. See, in agriculture, we are talking about the sustainable agriculture production system. And sustainable agriculture production system, it will be only possible when we will maintain all the agro ecosystem nicely, then only this production it will sustain. And it is not only the production, whatever we are producing, it is going to the consumer. And many rules and regulation, it is going to be changed. If consumer is buying anything, he want that the origin of the product, the residue in the product, the protein quality, and uh, many other things that can damage to his health. So everything he want to be written on the package itself. So I mean say that we have to increase the biofungicide, and we have to decrease the synthetic fungicide in the phase manner, so that we could come to a stage where our production system, it could be sustainable. Our consumer, they should get the best product. So now you will see, I mean say most of you are the student. It is very simple things, and you know the fungicide. The main function of the fungicide in the very simple way, it is going to reduce the growth and reproduction of the pathogen and any fungus. Fungicide is very simply playing a action that either it will kill the fungus, either it will inhibit the germination of their reproductive structure like spore sclerosia. It will not allow the pathogen to grow and reproduce. It will. So this is the major action. This is the major action of any kind of fungicide, whether it is a synthetic fungicide, whether it is a bio-based fungicide, whether even we are talking about the resistance gene function also, that is already, I mean, say breeder, they are used to deploy. So all they are concentrating only just to reduce the growth as well as the reproduction of the pathogen. See, very simple concept of the fungicide is, if I mean, say very simple. Every student, they might have realized before the COVID situation there was no COVID, but once it has been introduced, then it has started spreading. The same concept with the fungicide or any biomolecule that we are using for the inhibition of the fungus. It is very simple. That it is very simple. Inoculum is directly proportional to the disease. Inoculum is directly proportional to the disease. If there is no inoculum, there is no disease. If there is less inoculum, there is less disease. There is more inoculum, more disease. So what we have observed during this COVID period, before COVID, there was no inoculum, no COVID. When we have introduced the inoculum initially, inoculum was less people they have infected. When more inoculum, more people they have been infected, disease it has spread. And then it is now it is an outbreak, global pandemic. It becomes the global pandemic. So the main concept what we are using with the fungicide, we are trying that inoculum through this chemical and biomolecule, we want to keep the inoculum near to zero. If we are going near to zero, definitely I would say effort we are making. And what fungicide it is doing, definitely if initially it is used, then fungicide it is going to kill all the reproductive structure, those who are going to multiply. So the multiplication it is going to be reduced manifold because of the killing their primary structure. So there are two kind of inoculum, primary and secondary inoculum. If we secondary inoculum, it is originated from the primary inoculum. If you will kill the primary inoculum, because primary inoculum, they are very little, and they have been confined to the certain structure, certain places, and certain, I mean say, pockets. So there it is very easy to hit those pockets, those people, and those structures. But once it will spread out, it will be spread out and it becomes the secondary inoculum. It will be very difficult. Of course, fungicide it is taking care, but I would say that the pandemic or the epidemic, it is not going to be controlled. Now I am coming a bio-based fungicide. This bio-based fungicide, I would say that azoxystrobin, this is a fungicide, and it is derived from a fungus, Strobilurus cancellus. This fungus, it was growing in the nature, and what people they have observed that in and around where it is growing, no other microorganism, no other fungus, they used to grow. They thought that it is producing certain metabolites, that metabolites it is going to kill the fungus. normally the German scientists and the people from the probably from the Bayer group, that they have taken this fungus, they have came isolated the molecule, and finally they came to know that it is a azaxiestrobin, and it is going to block the respiratory chain, that I will come later on. And then what happened when they have naturally I would say that purified this molecule azaxiestrobin, it was not much effective. It was not much effective. So what they did, after knowing that this is the uh, this is the molecule that has been originated by a fungus Strobulus, and then they have made the, I would say, several thousand derivatives of the azaxiestrobin, several thousand, and screened all the derivative against the fungus. And finally, they came out with, out of several thousand screening. If you will see the patent, how many, if you will see their patent, you will see how many molecules they have screened and how much time they have taken for the screening of the molecule. And then finally, they strike one molecule that become commercial. So now azaxiestrobin, it is in the market. So many companies, I mean, say, so many other uh, kind of azaxiestrobin and its analogs, and I mean, say, similar kind of pecloestrobin, azaxiestrobin, so flucoestrobin, so many estrobin. It has, I mean, say, that molecule, it has been patented by the several companies. But if you look, this was basically the whole idea. It came from the fungus Strobulus. Now I'm coming this molecule. Now see this molecule. Now you can imagine the global market size to reach $1,024 million. I would say around maybe 10 billion. So this is single molecule that is doing, this is a single molecule. It is doing the business of around $1,024 million, and it is going to be more. It is going to be more. Uh, so, see, azaxiestrobin. Just I have just read my slide, and you will find this the chemical structure of this azaxiestrobin. I don't know much chemistry, and most of you are those who are much interested in the chemistry. They can go through the various kind of structure that I would say that Bayer company initially they have derived and tested it. So you can find out that what are the difference in the structure and the functional group, so that you can see the where this efficacy and how this molecule it is going to act. So now I will just I am showing you this slide, how this fungicide it used to work. This fungicide, it is a QI inhibitor, there's a quinone outside inhibitors. If you most of the biology student and I mean say chemistry student, they are knowing the respiratory system in the mitochondria and how the electron chain it used to, I mean say, start, and different kind of energy it is going to produce. So during the I mean say the process of the energy generation, this if you will see here, I'm talking about this cytochrome BC1 complex is there. The cytochrome BC1 complex. So the azaxiestrobin, it is coming and it is going to block the cytochrome BC1 complex. And when this BC1 complex it will be blocked, then respiratory chain it is going to be disrupted, and here it will be oxidative burst. It will be oxidative rust, and the membrane of the mitochondria, because of the oxidative rust, because electron coupling it is not going to be there, oxidative rust will be there, and it will be membrane, it will be damaged, and finally I would say mitochondria, that is has to be called the powerhouse of the cell. The cell it will get killed. So this is the what exactly I am going to convince our most of the student and participant, that see, when you are designing or you are going to have any kind of biofungicide, you must be very clear that what are the various metabolic pathway and which particular metabolic pathway you are going to target. I mean, so that what is happening most of the paper when we have reviewed, What is the difference between the university laboratory and the college laboratory and the industry laboratory? There is a big difference. Here, what we are doing, we will collect 100 plant and we will just get a extract. We will go for the in vitro test, and if growth inhibition is there, we used to say yes. This is the fungicide. No, unless until you are going to explore the entire process, how it is going to inhibit the cellular mechanism, cellular metabolism, then you can come out, we can modify. So like azoxystrobin, see, they have synthesized several thousand of the derivatives, and after that they have started screening, and finally they strike one molecule. Similar way, I mean say that thousand and thousand paper published in the various laboratory to come out with the biofungicide, but none of the biofungicide, it came, it became commercial. Reason was very simple. We have not given enough thought, but I would say the young mind and young people, those who are in the university system, those who are coming to innovate many new things, they can start thinking that we have to understand the entire, I would say, energy generation system. And see, the slide which I am projecting, there are succinate dehydrogenase, there are cytochrome BC1, there are cytochrome E oxidase, ATP synthesis. These all the target we can block. We can block provided whether we are coming, I would say that through the chemical process we are going to derive a fungicide, or we are coming a plant origin, fungus origin, microbial origin, bacterial origin, algal origin of the molecule that is going to block this process. And we have, I expect there are several molecules that has not yet explored. So we have to think and test. But our testing procedure, I would say that I will talk. Let me come to the second molecule. The second molecule I am talking about, the carboxin. This is a synthetic molecule, but how I brought this slide just to show you that what are the different kind of, I would say that when we are going for the citric acid cycle, what are the various reactions that is taking place. So you will see here this uh, fluoroacetate. This is aconitase. So this is a, I would say that's this a molecule, but this molecule it is going to inhibit, but it is non-competitive inhibitors. So this molecule it could not become successful. Again, arsenate. Here this is also a molecule, and it is going to act as a ketoglutarate dehydrogenase. This is also non-competitive inhibitors. But finally, if you will see, this is the succinate dehydrogenase. Succinate dehydrogenase enzyme, it is going to be blocked by the carboxyl. This is the molecule. Now this carboxyl, it become a commercial molecule. So this is the this is what I would like to convince you, that see how this, I mean, it's a various metabolic process we have to inhibit, so that the process of metabolic process of the cell, it could be blocked, and then cell should be starved for the want of energy, or certain metabolite that has going to be accumulated in the cell as a shunt product, and finally it is going to create a lot of damage to the cell, and cell has to be killed. So this is the whole idea, that we have to think when we are thinking, planning for any molecule from the bio origin, then we have to think what should be the target, where we have to hit. I'm just showing the few known target, but there are several hundred other target also that I am not knowing. But as a young inventor, you can think and you can come out. I'm saying it's definitely. So this is the electron transport chain, that what I have told, that these are the molecule. I mean, so, and that is working at the different process of the electron transport chain, and they are going to disrupt the process of the I mean, say energy process, and finally cell is going to starve the start of the energy, and finally going to killed. The next slide is that There are many. I mean, so this area it is not yet explored much. There are many non-ribosomal uh, peptidases. They are the class of the peptide of the secondary metabolites. The secondary metabolites, I would say that they are having the great scope, and mostly secondary metabolite it is coming uh, from the plant origin. I would say the mostly plant origin, algal origin, actinomycetes, so fungus also. So there are a lot of secondary metabolite and secondary product they are coming, and all these products they are also going to, I would say, inhibit the several, I might say that protein synthesis and many other activities in the cell. So I would say that these are the I would say that secondary metabolites and the non-ribosomal peptides. They are having the better scope, and how we are going to design it as a fungicide, that we have to think. Uh, then I would say that see, I'm talking about a fungicide. Most of I must say the girls student mostly they are using many cream, and this all the cream. Those whitener, I would say that cream means I'm talking about the whitener that is, I would say, keeping our skin much white. So this is all they are carrying an inhibitor. That inhibitor we will call it melanin synthetic inhibitors. So the melanin synthetic inhibitors, it is a uh, I mean say that this all these those inhibitors they are I mean say that uh, working at a different point. The whole process is the I mean so sicolin and sicolin. If you will see how the melanin and how this inhibitor they are going to be I mean formed. So if you will see the cell membrane, I'm talking about the cell membrane and mostly in the fungus. How a fungicide, because most of the metabolic process that I have talked, it is a common in most of the living organism. But where is the striking difference between fungus and between plant, between fungus and between animal, between fungus and between human being? So we have to be very selective. So the molecule that we are going to design, it has to be based on the certain selective function of the fungus. I'm talking about majority of the fungus. They are having in their cell, cell membrane, mostly the human being and others, we are having the, I mean, say the sterile. That is that I'm not able to recollect. Just forget it. But I mean, say that. But fungus, they are having a unique esterol. Esterol, it is a kind of fatty acids, and that and it is going to form the cell membrane. So it is ergosterol. We are not in the human being. We are not having the ergosterol. We are having the different esterol. In the animal also, we are having the different esterol. But ergosterol, it is very common in majority of the fungus. So what is going to be happen? We know this ergosterol, it is going to be synthesized by a process that is called the uh, succulin, and then succulin epoxidase, then it is going to be converted into the lanoesterol, and then for dimethyl cholestra 814 trienol. So like that, and this is the entire process of this membrane formation. What is going to be happen if we are going to damage the membrane of the fungus cell membrane? Then entire solute of the fungus it is going to be leaked out, and ultimately cell will not retain I would say retain its identity, and it is going to be killed. So, so now just to see the here the process, the lanoesterol. When lanoesterol is I mentioned that, if see succulin epoxidase here, they have designed the just to block the the synthesis process of the ergosterol. Here succulin epoxidase, it is going to be, I mean say, inhibit by the alamines. This is an effective inhibitor, and if you will use, then definitely the membrane, this ergosterol membrane, it is not going to be formed, and cell, it will be very weak, and most of the content of the cell, it will be leaked out, and cell will get killed. Then if this cell, it is going to be proceed, then lanoesterol, it is going to be formed. In the lanoesterol, see, there are thousands of the different azole molecules they have discovered. All these azole molecules, they are going to, I would say, they are going to inhibit the demethyl, I would say, lanoesterol 1,4-demethylase enzyme, and ultimately the ergosterol, I would say, membrane synthesis, it is going to be interrupted in between only. Now, I'm just coming just to see the another molecule, that is the morpholin. Morpholin enzyme, they are working esterol G8 I would say that esterol G4 reductase, and mostly the entire process. Some of this process, it is not yet known, but ultimately 4,4-dimethyl gymoesterol, it is coming to the phacoesterol. So in between this process, there are several molecules they have identified. They are belonging to the morpholin group of compound. So they are going to inhibit the membrane synthesis, and ultimately ergosterol, it is not going to be formed. If it is not going to be formed, then what is going to be happen? Ultimately, cell is going to be killed. So a large number of, I mean, say, azole molecule, it has been discovered. So now my point is how we are going to capitalize this information by our young mind and young people. Can we, number one, it's a very simple question. Can we discover this kind of molecule of the plant origin? Can we think, I would say, if this process is known, can we think this molecule of the plant origin? So these are the process why I have brought this information. Hello. I have brought this information before you. You can widen your thinking. I mean, you can widen your horizon of the thinking, because these pathway, they are the known pathway. So the very simple thing is, can this synthetic fungicide can be replaced? Process is known, and we have to think about it. The my next slide is, it is a what I was talking about, that the whitener, I would say that cream that we are using for glowing our skin and making is more white. So it is a melanin synthesis. So all those molecules, they are going to inhibit the melanin synthesis. So the most of the, I would say, earlier, if I have studied that some these molecules, they used to, I mean, say, purified and good kind of a product they used to put into the cream also. I don't know exactly. Now they are using the kojic acid because of the certain regulation and others. But this entire process of the melanin synthesis, it is very simple process. It start from the pentaketide pathway, and the first product after I mean, say, pentaketide pathway is the 1368 tetrahydroxynaphthalene, THN. THN, it is going to be then converted into cytalone, and then cytalone, it is going to be produced, it is going to be reduced. So 138 trihydroxynaphthalene, and then it is going to be the vermilon, and vermilon, it is going to be the DHN. I would say that DHN 1,1 dimer, and it is going to be polymerized, and it is going to become a melanin. So most of the fungus, those who are, I would say, if you cultivate and if you will see the fungus, majority of the fungus, they are having the melanin. Melanin is the most important molecule that is helping fungus to protect from the radiation, and just to penetrate. They are putting around, I would say, enormous pressure on the epidermis of the plant, and then they are going to be break out, especially the classical example is the blast pathogen of the rice. It is after germination, it will produce the penetration plate, and it is around, I would say, 8 kilopascal pressure it is going to generate on the epidermis, and epidermis will break out, and then fungus will get entry. But this entire pressure, it is managed by the, it is entire pressure of the pathogen, it is managed by the melanin. what melanin is doing, melanin, it is going to be deposited around the penetration peg, and when the pressure, hydraulic pressure, it is coming, generating in the uh, peg through the glycerol, and then that penetration peg, it may burst. So the melanin, it is providing a strength to the fungus, and then it is breaking it. If you will make a mutant, I would say that melanin synthesis process in the pathway, either you do the pathway, just close this pathway, melanin pigment pathway at the cytolone or at the bermelion level, you will see the pathogen, it will become white. It will not be black. It will become white, and it will lose its penetration ability. So what Japanese scientists, when they came to this information, immediately after knowing to this information, they have started synthesizing the molecule, and they came out several fungicide on this cytolon inhibitors. Some are the 73 tetrahydrophthalate inhibitors, then bermellone inhibitors, and then DHN inhibitors. So the 4-5 molecule they have designed, all it has been designed in the I would say that we used to group it, that EBI group of the fungicide, ergosterol biosynthetic inhibitors group of the fungicide, and this group, there are several molecules. Chemically, they are different, but the function is same. They are going to act at the different, I would say, stages of the melanin synthesis. So we know that there are many other. I mean, say, why I talked about the whitener and the cream. There are several. I mean, say bio-based molecule. It has been introduced in the whitener, and mostly they are also reducing this melanin synthesis pathway. And we can. I mean, say there is still scope. We can explore this pathway. So. Uh, uh, then, uh, then we are having many other inhibitors like glucan synthesis inhibitors. Mostly, if you will see the cell membrane, I mean the cell wall. So cell wall in the fungus, they are mostly the chitin. And uh, if we are having, I mean say that chitin and then beta-1,6 glucan and beta-1,3 glucans are there. If you are having any glucanase inhibitors, so we can able to kill the fungus. So that is the idea. So now there are I would say that the certain bacteria that our group we have discovered and published paper very recently. It is a halotolerant rhizobacteria. What exactly this bacteria it is doing? I would say it is not exactly a fungicide, but this bacteria we have I would say that from the sea origin it has been discovered, and it is controlling the Aspergillus flavus, and mostly Aspergillus flavus, when it is a producing a toxin that is called aflatoxin. It is the most dangerous toxin, and it is I mean say listed in most of the food stuff. It is a I mean say with the red means it is carcinogenic and very harmful. So this entire pathway, this bacteria, when we are using with the Aspergillus flavus in the peanut, then what we have found, it is going to induce the pipacolic acid pathway. This pipacolic acid pathway, when it is going to increase and induce in the plant itself, then the fungus growth, it is going to be stopped. It is not going to kill exactly, but definitely it is not allowing fungus to grow. So it is going to, I would say that it is what I have told in the initially, that either we have to kill it or we have to reduce drastically the growth and reproduction of the pathogen. Exactly this, when we are inducing the pipacolic acid pathway by using this halotolerant bacteria, then definitely this pipacolic acid, it is going to induce, and the growth and reproduction of the pathogen, it is going to be closed. So this is all about the pipacolic acid, and how we have done the metabolic profile and all those kind of things, and we are designing how to produce it. And see, this my next slide is I'm going to say some intervention, and that it is an insight that yes, we when messenger RNA it is going to be formed, then we have to design certain molecule, because we are knowing several interference in the messenger RNA. Of course, by I mean say that small siRNA or the miRNA, and I mean, say, very recently I have read one paper. It is coming by the Syngenta group. Probably they have designed the uh, I mean, say that the mRNA-based fungicide, and they have declared that they are coming in the market. When they will spray, it is going to block the I mean, say that protein synthesis in the fungus, and ultimately fungus they are going to kill, and they are claiming that it is a green molecule and it is going to be the most successful. So now I am coming to the screening of the fungicides. What is the problem and how you have to proceed? In most of the cases and most of the laboratory, we are having the two protocol initially for the screening of the fungicide molecule. In vitro, we are having the food poison technique and spore germination. Food poison technique we are using only for those fungus, those who are not producing the spore. I mean, say it is only to the mycelial state. And second one is the spore germination. So those who are reproducing through the spore, we are looking about the spore. I mean, so how we can inhibit or how we can kill the spore. In the food poison technique, what exactly we are doing? We are having a medium, and that medium should not interfere the molecule that we are incorporating. We will incorporate the different concentration of the molecule, then we will inoculate the pathogen, and at the same time we will have a control check where we will not put any I mean, say test molecule, and we will compare how much growth it is going to be reduced. Similarly, in case of spore germination, we will go use the different concentration of the molecule, and then we will see how many % of the spore it has been germinated. So these are the very simple techniques. But this technique, it is not, I would say that the end point. It is just the beginning. But most of the paper, what I have seen, thousand and thousand papers from the various laboratory, they are concentrating only this, and they are claiming it is. I would say it is just the beginning. Uh, just to screen the thousand and thousand molecule, because these are the very simple technique. But this technique, it is I would say that not very I would say that not very uh, 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 giving a complete insight that your molecule it is likely to be a future biofungicide or future fungicide. No, in the what is the problem with the food poison technique? Because in food poison technique, you are creating horror in the plate against the pathogen. I would say that it is a very clear-cut concept, and you are not knowing what are the osmotic balance of the medium and what are other factors that is going to interfere with the growth of the fungus. So that we are not knowing unless until we will make the every factor constant, then we can come out with the I would say the fungicide. But food poison technique, it is giving an indication. If you will repeat your experiment times and again, you will find, yes, it is a giving the consistency. And if you will use the different kind of pathogen with against the same molecule, then you have some insight that yes, certain things are certain molecule, it is there, and we can proceed it. And then similarly, in case of spore germination, also, if once you will screen the thousand of the different, I mean, after repetition of the different 100 times, you will become sure that yes, certain molecule are there and it is really inhibiting the growth of the pathogen. Then you have to try to make it. You have to try to purify it. So there are several purification techniques. Now it became a chemical jar. So once you will purify, then you have to, I would say, we use the purified molecule because here it will be a direct interaction. In the crude, there are several other things, several different kind of things are there, those who used to interfere with the growth and reproduction of the pathogen. But once we will get a purified molecule, then definitely it is going to help us that yes, it is the actual molecule. And after knowing, after coming to know that this molecule it is effective and really it is having the fungicide value, then we can go for the structure and we can look about the molecule configuration. And through this configuration, uh, we can find out that how and what are the various metabolic pathway where this molecule it is going to influence the pathogen. Similarly, in case of spore germination also, because the both the function, either the growth inhibition or the spore germination inhibition, it is because of the certain molecule that is going to be blocked somewhere. So my point is, now you just see I'm presenting the different kind of I have picked up this slide from the Nature review of the microbiology, and these are the different kind of I would say secondary metabolites, and these secondary metabolites, they are effective at the different I would say conditions, and as well as they are having the different kind of I would say function. So uh, if the secondary metabolites, they are going to I would say that disturb the different kind of function, then we have to definitely discover this so many new secondary metabolites, that what we have discovered the picoleic acid. So I would say that discovery, it has to be made, and then we can understand, because after the whole genome sequence of the various pathogen, I mean, it's a prediction now, it become very easy, that what are the different kind of function it is going to be blocked by using certain molecule. Uh, yes, then I am coming to the, see how this is a I would say ergosterol bias, and I would say melanin inhibitors. Now see, if we are using I would say the melanin inhibitors, then what is going to be happen? The same fungus, it was a control, it was a black, and when I will use the, or you will use the melanin inhibitors entire fungus, it has going to last the melanin, so it become white. If once it become white, then many process, because melanin, it is giving lot of strength to the mycelium, and then the process of the asexual reproduction, it will be drastically inhibit, or it will be inhibit I would say very much. And just see this slide, what is going to be happen when here the I would say tricyclazole, that azole group of the fungicide, when I am using, if it is not there in the medium, then all the spore and everything, it is going to be produced. When we are using a little 100 microgram this azole group of fungicide, what is going to be happen? Just see the development of the conidia and conidiophore. That is the reproductive structure that is coming out from the vegetative growth. It is not going to be formed. It is going to be poorly developed. And once it will be poorly developed, then the spore, that is the real I would say the culprit for the spread of the disease, then they are not going to be formed. And growth of the pathogen, if you will see, it is also going to be reduced. So only by melanin, because melanin is needed in the conidiophore and the conidia for its stem and for the morphogenesis of the fungus. So I would say that what exactly we have to see, that I would say, if we are using any molecule, I mean, say for example, if I have gone through the in vitro I would say that in vitro test for the in vitro growth test, then we have to see what is the function of that molecule, whether that molecule, it has brought any changes in the growth biology or the reproduction biology of the pathogen. That most of the people, they are just going to take the, yes, it has inhibited. They are not going into detail that yes, if it is going to be going to inhibit the fungus, then what are the function that is going to be inhibited? So mostly the entire inhibition process, either the initial growth, it has to be checked, differentiation of the mycelia, it has to be checked, or the reproduction system that is coming after the growth of the mycelia, it has to be checked. So we have looked, I would say, that different stages, how a molecule, it is going to affect the growth as well as the reproduction of the pathogen, and that what I have shown here. In this slide, you will find everything intact, but here you will see the melanin, it has been lost, and then again you will see there is no conidia and conidiophore development, and the growth, it is very poor, is sparse, and no conidia and conidiophore or reproduction system, it is going to be there. If you will see about this I would say these two conidia, conidia initially, it is going to be formed, they are not going to be developed further, because conidiophore, it has to erect in the air, in the plate itself, but it has to come in the air. When it is coming in the air, the conidiophore, it is not having enough strength to bear and to become erect. So it is going to be collapsed because of the lack of melanin. So I would say we have to have an entire vision. What are the various biological process and biomolecule that is present in the fungus, and that it is going to be inhibited, and it is not going to be formed. So, and after that, okay, I agree that yes, you have come out successfully after doing all these things, and you have synthesized, you have purified a molecule, you have got the structure and everything, and uh, you came to know the function. And then after that, because it was a in vitro test, I will say, now you have to go for the part experiment. But in the part experiment, we have to have a certain design, because now the now here the chemical as well as the environment interaction, it is going to take place. In vitro, everything it was controlled. You have not any, I mean, so much variation. But the moment you will come for the fungicide, it is going to be used in the field and on the animal and in the different environment. So many things are there. So we have to see how much environment it is going to interact. Then we have to have certain design. For the initial, we are going for the part experiment. So we have complete randomized block design and minimum number of the plant and minimum number of the part and plant. We have to ensure the reproducibility of the result. Whether, I would say, how much deviation it is coming from part to part and plant to plant. So all those things we have to know in order to have a robust idea about the molecule. So, and then we have to ensure that pathogen it is there, and I would say that these things we are taking care by having a control plant. When we are having the control plant and we are not inoculating the pathogen and all those, and then comparing the things with the, I would say, inoculated plant, and then we are comparing the result and the data recording. Data recording is the most important, because if you are not having very data from the very beginning, you are going to file a patent, you are going to, I would say, claim. You cannot claim it unless until you are having all the data, methodical data, your diary intact. Then only it is possible. Otherwise, you will lost somewhere. So for the young, I would say, student and young innovator, I will say yes, they have to keep all the, I would say, data and their diary intact, so that whenever you are writing for the patent or going for the IP claim, you produce all the record. And then it is coming the field trial. Field trial is very tricky, and there are certain standard. And I would say that I feel if you look about this molecule that I'm talking about, if a molecule discovered in the US, if a molecule discovered in the Germany, it become a global product. How it become a global product? It was reason that they have tested all these molecules in the different across the countries, across the geography, across the environment, across the time. They are testing, and then that all those products it become the, I would say, global product. So because at the time, at this space, at the environment, it has proved its efficacy. That's why they are earning the billion and billion dollar. Like Ajax yesterday, that one molecule, what I have told, that Bayer, it has more, it is earning more than the $1 billion. Reason was that it is effective everywhere. We have also recommending to the farmer, we have also tested as on the different crop communities, and we have found it is an excellent molecule. So I mean, say that we must have a concept and idea how it is going to be a product and a commercial value. So the field trial is the most important. In the field trial, we have the randomized block design, then standard plot size. I would say most of the data, yield and many other things we have to compile. Then it has to be replicated trial. It must have the 20 square meter field size, then presence the pathogen, and then data recording and analysis. So these are the things. These are the things, and what I have told. That's my point is this how the this ergoist, I mean, say that melanin biosynthetic inhibitors, it is germinating. How it is just see here. These are the melanin deposition, and it is penetrating to the epidermis around 80 kilopascal pressure. It is going to generate to break out the epidermis. So And then the multi-location and multi-year trial that already I have discussed you how a product it is going to be a global product. If it will not be multi-location and multi-year trial, it will never be the it will never reveal you the effect of the fungicide or your molecule at different level, and because environment it is going to interact everywhere on the molecule, and so we should know the efficacy of the molecule, then phytotoxicity, and environmental data. These are the I would say that the very much essential, and then so these are the things. But I would say in the university or in your laboratory it is not possible. It was exactly I am going to convey you. You have to come to the first stage where you have come to the part stage and you have to come to your field stage. Then the molecule for the commercialization, now it has to go to industry, because the data, phytotoxicity data, it will take several crores rupees. It will need it. Phyto, because your trial it has to be conducted. It will see the regulatory authority and all those. They will ask the huge amount of data to get it registered. And even if it is a biofungicide also, if you the moment you will say the fungicide, then you will come under regulation, whether it is a bio origin or it is a synthetic origin. So normally, if you are confident, then you can negotiate, you can file a patent and then start negotiating with the certain companies, multinationals, all those they are there. They are looking for such kind of green molecule. So you can collect, and remaining test they will do it, because it is a huge investment. It is a huge investment only for the phytotoxicity and environmental data. At least in one country, 2 to 3 crores it is needed. So uh, I would say that uh, this is the process how we are coming to this. I would say the biofungicide. But if you are clear, don't dishearten. If and don't be discouraged. What I have told you, you must be the bold and robust with the knowledge, and see the many companies and the many inventor. They are coming like that. They are purifying their molecule, they are testing their molecule, and then they are filing the patent, and then they are negotiating the company. So I would say that scope is very good. Yes, you have some bypass also. If you have a I would say, if you want that yes, this molecule it is very effective, then I would say that it is not I would say the correct process. It is not ethical also. But initially, I can just to encourage you that yes, you can have some. You can just name, but without disclosing your chemical property or anything. I would say you can commercialize as a bio stimulant, bio inhibitors like that. That I would say it is not under regulatory control. But the moment you will say the fungicide, the moment you will discover the chemical, but the moment you will claim, then definitely the regulation it will come, and they will not allow you to commercialize it. So this is the I would say that the process about the how you are going to discover a molecule, and then how you are going to I would say that getting it registered, and getting it joint venture with the private companies, and are alone as a biostimulant, as a bioproduct, how you are going to commercialize it. So this is up to you. So now my talk is over. If anyone want to interact, certainly uh, I will answer the question my level best. Thank you.
Thank you, sir. So students, now quickly, we have 10-15 minutes to discuss the queries. So yes, please.
Hello, sir.
Yes, please.
Good morning, sir.
Good morning.
Sir, my question on my grassroots level. There
Yes.
is, sir, a mango plant I
Yes.
crop in my garden.
Okay.
So, sir, 2 years ago. But,
Mm.
sir, my mango plant can't be, sir, growth on the like any fungus attack, I feel, sir. So, what can be happen, sir? I mean, what is the reason behind the why my plant is not growing and some leaf are not freshly, sir, growth, sir?
I
So
would
is
say,
the sir fungus attack or sir any other disease like sir?
see, if your leaf and everything it is clean and there is no any, I mean say, blemishes or any spot or anything, I would say that if there is no necrosis, cell is not killed, then apparently I feel it is a healthy plant. Now you come to the root, I mean it's a below ground plant that is not visible. So we have to see and see the I would say that you might be having the soil survey and land use planning data. Is there any deficiency in your soil, or uh, uh, um, are some fungus? It is I would say certain pathogen, nematode, and all those insects. Some are there, those who are damaging your root. So you have to see. You have to see, and the best thing is that normally what we used to recommend that you give the balanced fertilizer and put some organic matter. So what exactly it is going to be happen when we are putting the organic matter, lot of microbial activity it is there. So they used to take care of the root pathogen. So I would say that is my suggestion.
Okay, sir. Root care, sir, pathogen, sir. Right, sir.
Yes, yes,
Okay, and sir, my second question, sir. Sir,
yes,
you told the sir bioresources like fungicide,
yes,
like sir white mushroom, sir. You told sir,
yes,
in which the sir new molecule, sir, scientist
yes,
has sir discovered. So
yes.
sir, I want sir another sir fungicide example of the bioresources, sir.
Uh, um, bioresources. Then uh, I don't have, I would say this was the Ajaxius to bean only. That was from the bioresources. And most of them other are the, I would say synthesized. But insecticide, it was the pyrethrum. That was came from the pyrethrin. Pyrethrum.
Okay, sir, pyrethrum,
Yeah,
sir.
pyrethrum, it is an
Okay, sir.
insecticide, but
Thank you,
lot of
sir.
derivatives, and delta methrin and all those, they have synthesized. But it originally it was the concept, it came from the pyrethrum.
Okay, sir. Hello, sir.
Hello, hello, hello. Hello.
Hello, sir. You hear me, sir?
Yes, please.
Actually, sir, your voice is sir
So
Ah,
terminating, sir.
no, 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 no.
Hello.
you should quick in your query so other students can chance. Any other student? So, sir, I have a question.
Yes, please, please.
Sir, what need to be targeted according to your suggestion? Like we should target the microorganism or we should go for the bioactive compounds?
See, it is not the microorganism. Really, it is a bioactive compound. See, if now in the genomics era, the many molecule it is produced by the from the bacteria to the human being. It is a, I would say, so it is the only the metabolites and the compound that is going to be effective. It's not as such. Otherwise, if you are using the microorganism as such, then it will be competition. It will be a amensalism. It will be commensalism. There are several kind of. I would say that is a different issue. But when we are talking about the fungicide, then definitely we have to target more about the molecule, and that has to be the active molecule. So bioactive molecule definitely. And target is also known in the fungus. If you look, most of the fungus genomics is known. Functional it's a function is known. So the part that I have presented. So I would say that you can ask some bioinformatics people, and they can see. I would say they can go further. I would say that in silico test, and they can indicate initially that these are the I would say bioactive molecule, and these are the target. It is going to likely to give the result. So our process and our time it will be reduced.
Okay, sir. So I thought there are a number of biocontrol agent which is approved by government and those are in the market now. So I think, sir, they are encapsulated or maybe formulated by using some microorganism, right, sir? Yeah.
So the Srishti, I am looking the canvas beyond that.
Right.
See, see, all the time we cannot sustain because the I would say that the microorganism formulation and still we are not knowing exactly how they used to do. Only we are knowing the result. So the what is going to be happen? The IP that we are targeting, the business that we are targeting, it is not likely to come because most of my friend I always used to oppose them because I'm working on the resistance, I'm working on the gene and genetics and genomics. So I always talk them that you talk about the molecule, don't talk about the microorganism. It's because you are in the university, let the microorganism, it is beneficial definitely. It has to go to the farming community. But as a researcher, we have to promote the IP, we have to promote the industry, we have to promote a new innovation. And this innovation, it will be, see, if someone is testing, ultimately what is happening, you have isolated, I mean, it's 1,000 different trichoderma, we screened it, and then gone for the field test, you are saying this is the X molecule, it is effective. X species, it is effective. Why it is effective? What are the conditions? What are the molecule? Because ultimately it is the chemistry, it is the bioactive compound that is making the hell of difference. So that differences, if you will not know, so we can't sustain. So my always I used to emphasize, like in plant breeding, we are discovering lot of genes every day. And see, this is also a kind of plant protection mechanism. So we are discovering, we are pyramiding, and we are putting together in a block. So ultimately, we are knowing that what are the function of these genes, how what are the different molecules they are going to produce. We are knowing, but why the biocontrol people they are not doing it? I mean, so it has to be parallel. It has to be parallel, and they have to come out with a good science and then good molecule and good IP.
Right, sir. So I think many of the scientists and many other innovators are running behind finding the novel microorganism to
Yes,
make a biocontrol agent that is completely, I mean, that is not completely, but that is some way very, specific way. Instead of that, we should target the particular bioactive compounds from microorganism. That
definitely,
I
definitely.
agree.
They will introduce the novelty. I mean, so we are looking for the novelty for the entire research.
Right. So again, if we use a microorganism, then that question comes of that commercialism and again the competition between the indigenous microbes as well as the biocontrol agent. Right, sir. I agree with you. There are many marine organisms which are not targeted, I think yet. Like for example, actinomycetes or we can say the microorganism from the deep sea, I can say.
Yeah. So
So
the sister, you see the I mean, say the Alexander Fleming, Waxman. How I mean, so these people initially what we are thinking that they are reducing the growth of the fungus and all those kind of things in the plant. They realize it, and still we are okay. We are capitalizing it. No harm. It's a good thing. But why the Waxman and why the Alexander Fleming? They were the successful. Reason was that they looked beyond that. They thought that if it is in eating, certainly there may be certain molecule. They discovered the penicillin, they discovered the actinomycetes. So I would say that whatever you are saying, the sea marine microorganisms, they are really the treasure, and we have to hunt that. And mostly for the bioactive molecule, I would say that what is happening, why we are not successful because our team is very weak. We are not we are not making a good team of the different university and intelligent people, different research institute working together and come out with a molecule. That is the problem. If you will talk, yes,
yes, sir, teamwork is very important and
very,
we feel that in our country it is lacking because
yes,
many scientists together cannot work because of many strategies and many rules regulation of the central university and central labs. We can totally understand, and what this is what we are facing in COVID-19 as well, because there are many scientists. I mean, there are very few virologists in our country, and they can't work together to come out with some solution. I can understand, sir.
yes.
Yeah. So, sir, thank you so much. It was really informative. And Sorry, if anybody has to have one or two questions before we end. Quickly.
Hello, good afternoon, sir.
Good afternoon, Nilanshu. Okay. Good afternoon.
Sir, I
Nilanshu.
have a question.
Yes.
Sir, do lipopeptide affect the pathogenicity of powdery mildew pathogen?
Powdery mildew pathogen, lipopeptide.
Yes, sir.
Hello.
Yes, sir. Yes, sir.
So definitely, I would say that I have no such information, but the powdery mildew, mostly it is a ectophytic fungus. So lipopeptide, I would say it is, and it is ectophytic fungus, and mostly they are Hastoria they are producing. If any molecule, it is going to reduce the Hastoria, and penetration of the Hastoria, definitely the fungus, it will not be able to obtain its nutrients and it will be killed. So if lipopeptide, it is having such kind of property to just to, I would say, either to germinate the conidia of the, I would say, powdery mildew, or it has to inhibit the formation of the penetration peg. Otherwise, I would say that definitely, if it is having such property, it may affect. But some, I would say,
Okay.
I don't know exactly.
Okay, okay. Thank you, sir.
So, sir, there is a small question in chat box that one student is asking. We have grown curry plant in our kitchen garden, and
Okay.
white spots on the leaves is the major issue. His father sprayed many pesticide, but it is of no use. So, how to treat that?
See, seriously, it is not white mold. It is a insect that is called mealybug. It is not a, it is a mealybug. Just ask him. I would say, take the forcible, I would say, a pipe with the water and wash it. And continuous wash, don't allow them to grow again. It is mealybug, and that mostly it is a fly. It will come, lay the egg, and then a powdery mass, it will be there. So it is, I'm frequently, I mean, I'm doing the rooftop cultivation, so I'm frequently facing. So every day morning I am going, washing all the leaves and then coming back. So I have controlled it. So it is very easy. During rainy season, it will not be there because rain company, I would say, occasionally it used to wash such a, I would say, such insect where they have laid their larvae, it is growing there. It is not fungus.
Right, sir. So I consider there are no more questions. And student, if you have any question or query, or if you want to discuss your projects or any dissertation work you're doing on this regard, you can send me a mail or you can drop down the message in WhatsApp group. I'll definitely convey that to professor. So sir, it was really informative and very interesting, as I have submitted one proposal on biocontrol agent from marine microorganism to the DBT. Unfortunately, they have not accepted it, though they have not given any technical reason behind rejecting the proposal. But now I have clarified many doubts by your presentation. It was personally for me very useful. And yeah, sir, thank you so much for your time.
Thank you, Srishti. Thank you.